Hello everybody. I'm just going to quickly run through some of the lecture content and just give a bit of an overview of some of the key concepts and some of the things that we were talking about. This is not meant to be a crash course in molecular biology. A lot of what I say you will need to reference the original lectures and videos to really get the context and the understanding, but I'm just kind of summarizing a few key points here. Okay, but a lot of these summaries don't make a lot of sense unless you know the information that I am summarizing. Okay, so it's important that you go back to the original lecture content, and your primary source of study is the original lecture content. But having said that, I'll just quickly whip over some of these slides and just talk about some of the concepts. So the first lecture was on predominantly colon cancer. And towards the end of it, we talked a little bit about metastasis and angiogenesis. So in this series of lectures, we're using colon cancer as a primary example. And we talk about, there's a lot of things we can understand from colon cancer that apply to all cancers. So it's a good model and it's been well studied. So there's a little slide example here showing an image of these villi and on the lining of the colon. And just to give a bit more clarity to that, I included this slide here, which shows a profile of one of these little these crypts, and it highlights these stem cells at the base, and these so it highlights at the base here are the stem cells. And these stem cells are constantly growing and dividing, and when you get mutations in these cells here, they give rise to those mutations being propagated in these transient amplified cells. And as more mutations occur in the stem cells, then over time these short-lived transiently amplified cells, as they're constantly being replenished, as they die off and are replenished and die off and replenished, being sourced from the stem cells, then the mutations that occur in the stem cells end up being displayed in these transient amplified cells. So and that's a concept we talk about later when we look at clonal selection. So I thought it was good to include a slide that's not from the textbook that just talks to this slide a little bit. Okay, so we're talking about the accumulation of mutations within a cell, and about if it's a rapidly dividing cell, it's giving rise to other cells that have that same mutation. So we're talking about how cancer evolves, whether it's a single mutation or multiple mutations. And I think it's pretty obvious that it's multiple mutations, but when people started to think about this the first time, it wasn't obvious. So we talk about multi-step tumorigenesis, and we're just talking about this accumulation of mutations within a genome, within a cell. And we look a little bit about the time frame of this. I mean, is cancer something, if you suddenly are diagnosed with a cancer, is it something that occurred and developed over a short period of time, or is it something that's been developing over a long period of time? So we looked at some statistics and some epidemiology of cancers, and we looked at the incidence of lung cancer, and we were just noticing in these things here that you've got the occurrence of lung cancer over a period of time, and you can match that to the increase or uptake in smoking. And if this was 1930s in the 1970s, then the uptake of smoking is mirrored by the development of cancer. So this is clearly indicating a long time frame from the exposure to the occurrence of the disease, which gives a bit of a hint about what's going on. Also, I guess when you're thinking about this process, you can think of we know the frequency of mutations is quite high, so it's the accumulation of mutations over a long time period. I guess one way to think about this is we've got a large genome and mutations occur sporadically throughout the genome, and you need to have that, coincident of a mutation occurring in a gene that's involved in cell regulation, cell cycle growth or cell cycle proliferation, differentiation, one of those, apoptosis, one of those important processes. A key gene, eventually, as mutations occur, will pick up a mutation. Okay, and then we talk about whether those mutations are oncogenic mutations in terms of hyperactivating an oncogene, or whether those mutations are mutations that inactivate a protein. And then we talk about tumor suppressors. So we talk about loss of function of tumor suppressors and gain in function of oncogenes. And don't forget, we also talk about there were 2 alleles for each gene in the genome. With oncoproteins, hyperactivation of a single allele is rare, but is enough to give a change in phenotype with more growth. Whereas with tumor suppressors, there were, again, the 2 copies, 2 alleles. You need to knock out both copies because the remaining good copy can protect the cell from the loss of function. Okay. So again, you need to, when you're looking at oncogenes and tumor suppressors, you need to think about the genetics of them, 1 allele versus 2, and what's the consequence of having mutations in those scenarios. Anyway, we'll talk about that later when we get to that stage. We also talked about looking at various cancers which all follow this idea of over a long period of time increasing incidence. Again, this is going back to the idea of picking up multiple mutations over a period of time. Okay, we then went to look at the age of death from common types of cancers, and I think the take-home message there is that it's not related to it's not primarily related to your socio-economic group or where you live or those kind of things, but it's to do with the accumulation of mutations, and it's genetic, and there's a lot of similarity in the occurrence in different parts of the world. We also looked at the rate of progression, and if you're exposed to a mutagen, then the rate that is converted into maybe a cancer or tumor progression, it's the same regardless of whether you're exposed at a young age or an old age. It's the rate of progression is based on the accumulation of mutations driven by the mutagen. Okay. The textbook then starts to look at various biopsies taken from patients with colon cancer, and this is one of the advantages of studying colon cancer. You can do, colonoscopies and look at biopsies, and they started to look at the development of the cancer, and they started to notice the occurrence of polyps, and it turns out polyps were the early stage of cancer development. Okay, and they started to formulate this idea of a succession of phenotypes, thinking that maybe that over a period of time that we have this situation here where we have a tissue that's picked up a cell that's picked up a mutation, and because these are rapidly growing cells, therefore this cell is growing at a faster rate than the normal cells. So by the time this cell, let's make some general assumptions here, by the time this cell doubles to give 2 cells, this cell might have doubled to give, say, 3 cells, for argument's sake. So the rate of growth of this has been slightly enhanced by the mutation it's accumulated, and over a period of time these cells will outcompete the normal cells. So these cells will start to dominate the population, and that's what's being shown here. Okay, and then And then within one of these cells, which is originated from these cells, another mutation occurs. And that gives it a growth advantage over the surrounding cells. So this one then starts to dominate. How can we sort of represent this? Let's think about this. So let's assume we've got a biopsy of a tissue here, and they've detected an unusual cell here, okay, with a mutation in it, as opposed to the normal cells, okay. And over a period of time, that cell has a slightly enhanced growth phenotype, so it simply grows and divides at a faster rate than those around it, okay. And then within one of those cell types, it accumulates a different mutation, a different phenotype, okay. So this will now start to outcompete against the parent cell and the normal cells. So this one will start to grow and outcompete those around it, okay, and so forth. And this process occurs up until the point where the cell line has picked up enough mutations within it to give it a growth characteristic that, or other characteristics are affected in cell biology, so that it can begin to metastasize, and it can begin to release factors to draw in blood vessels to it, so that it can keep growing, and we've got nutrients and, you know, oxygen exchange, and at some point it's able to survive in isolation from those around it. And at that point, it's able to leave and metastasize to another part of the body. Okay, so again, in the lectures, we talk about some of the factors that control angiogenesis, some of the factors that are involved in metastasis, and some of the factors that are involved in the destination tissue that make it a good home for the new cell. All right. So again, there's a whole bunch of stuff in the lectures concerning that process. So this generally, this change in phenotype over time is referred to as a succession of phenotypes. Again, we're often looking at the same thing from different perspectives. So we're looking at mutations within a cell at a cellular level, and changes in mutations of DNA at a cellular level. But as that cell is growing and dividing and can be observed, then we see a change in phenotype. So then we talk about a succession of phenotypes over time, okay? And then as these phenotypes are things that we can recognize in terms of polyps and, you know, adenomas and all these late stage cancers, we can then talk about the evidence that shows that we're getting these multiple mutations within these cells. Okay, so and also we talk about clonal selection, which is again cells that have picked up mutations that have growth advantages compared to those around it. So in a lot of ways, it's similar concepts, but looking at different scales. All right, and if you understand a process at multiple scales, then you have a much better understanding of a process. Okay, so the other things we're starting to look at here. So this succession of phenotypes, it could have been other things that were going on at the time. So they had to prove that it was a succession of phenotypes. So using colon cancer, they looked for evidence that what they thought was the most logical process was actually happening. All right. So some of the evidence they've got here, I've got 3 points of evidence, first, second, third. So one point of evidence was that when you're looking at normal cells, so you've got these normal cells here, that at some point that there's been a mutation of some description, and there's a growth of cells here which are growing at a faster rate than those around it. All right, so this might be some small growth that's observed, I don't know, a polyp of some description or something. And then from within these cells, additional mutations have occurred, and then there's another growth here which seems to be emerging from the first mutation to give the second sort of observable phenotype, these cells that have got a different characteristic. And then likewise, this can give rise to another pattern of growth here. So it's a case of from the original normal cells, there's been a mutation leading to growth here, more mutations leading to the more severe phenotype, and more mutations leading to what may become metastatic or a problem for the body. Okay, so these were observed in biopsies taken from people with colon cancer. Okay, but to go from normal cell to the first stage might have taken a year or two to observe that growth, and then there might have been another stage that takes a couple of years to observe, and then another stage that takes years to observe. And obviously when you take a biopsy from someone, it's a snapshot. It's not something you observe over a 3-year period. So they were observing this phenomena, which makes sense in terms of the succession of phenotypes, but they're not actually studying and observing one batch of cells that are growing into malignant phenotypes over time. So you can't just say, well, I've observed that, therefore it's a succession of phenotypes. All right? It's a piece of evidence that lends weight to the hypothesis. All right? So don't think of things so simply, but it's a good piece of evidence. All right? There's a second piece of evidence here where they were looking at people. So they were taking colons from different patients, and if a patient has these little polyps that are starting to develop, then over time these were leading to full-blown cancers. Okay, the dreaded C word. All right. Whereas people with less polyps were getting less occurrence of cancers. Okay, so that is saying that over time these things must be developing into more full-blown cancers. Okay, just because they're present and over a period of time. So again, look at the slides for a more detailed description, but that's the kind of other evidence we were looking at. All right, and then the third piece of evidence was that there's a disease called familial adenomatous polyposis, so FAP. Okay, and as part of that disease, there's a gene called APC. All right, now this gene turned out to be a tumor suppressor, and it's commonly found in people with this disease, and therefore you can start to relate the occurrence of a single hit, a mutation in the disease, which is important for understanding multi-hit hypotheses and all of those processes. But also you can study those patients, and they have many more polyps in the lining of their intestines than normal people because of this mutation. All right, and then from these studies there's a higher incidence of cancer. All right, and again that's lending weight to the idea that people are more prone, and therefore it's a succession of phenotypes over a long period of time. Okay, so there's a lot more evidence out there, but the textbook highlights 3 pieces of evidence. So have a look at those bits of evidence and have an understanding of why they're important in looking at this, of solidifying this concept of multi-hit mutations, but in particular succession of phenotypes. Okay, so we're looking at it at a phenotypic level here. Okay, and then they studied this in colon cancer, and they also then started to look at other types of cancer, and it's a common theme. It's not something that's unique to colon cancer, but doesn't help us with the understanding of other cancers. All right, so I guess then the important thing is to say, well, we know it's the accumulation of mutations. We know one of those mutations is APC. We've got to understand what those other things are. Okay, what are those other things that are mutating? All right, and this led to the development of ideas about gene function. So we have this class of proteins that were initially discovered called oncoproteins. Okay, and RAS was the big one, SRC was a big one, receptor tyrosine kinases, they're big ones. So there are classes of proteins that have individual functions in the cell, but we can generally call them oncoproteins. And the characteristic of these things is that they can pick up mutations in one copy of the genome. So one allele of these oncoproteins can pick up what's called a hypermutation, and it makes it locks that particular protein in its on state. And that's a pretty vague statement, but if you look at an individual protein, you can understand what that mutation is that locks it on. Okay, and so if you look at RAS, you can look at which point mutations, knock out the ability of RAS to hydrolyze GTP. If you look at the receptor tyrosine kinase, you look at mutations that lock the receptor in a structural form that is constantly signaling. Okay, so for each protein, there are different ways in which these point mutations can lock things in the on state. And if you can do that to a protein, then it's an oncoprotein, and it's driving growth. Okay, so people started to think about oncoproteins a lot when they studied cancer, and then later on they discovered tumor suppressors, and that came out of the study of retinoblastoma, the disease. Okay, and they discovered a protein which they called retinoblastoma, and a gene which is the retinoblastoma gene. But an understanding of how that protein works is pivotal in understanding how many cancers work, even though it was identified in a disease of the eye, which is retinoblastoma. But it's also identified a new class of tumor suppressors. Okay, so then they had not only oncoproteins but tumor suppressors that have a role in cancer. Okay, and then we can talk about that as we get there. All right, so let's keep going. So when you look at the breakdown of types of mutations that are observed in these cells, they notice that more tumor suppressors were inactivated than the number of oncogenes that were hyperactivated. And that's because it's harder to hyperactivate a protein. You're relying on a single point mutation to enhance its function. And that's a very rare event, but it does happen over time. Whereas with tumor suppressors, any of your kind of bog standard insertion, deletion mutations are going to throw the reading frame of that exon, and it's going to that protein is going to lose its function because it's going to be inactive. Okay, and once you start to lose a tumor suppressor function, that's going to allow the cell to grow. And because those kinds of mutations are more common, then these tumor suppressors tend to outnumber the number of oncoproteins. We then start to look at so we've talked about this succession of phenotypes in a fairly generic way, looking at those cartoon diagrams. This is just showing a succession of phenotypes within a patient who has colon cancer. And you can see here that you go from a small polyp to a larger adenoma to a mass of these tumors that can become malignant. And we know that during the stages of that transition that we're accumulating more mutations, leading to more severe phenotypes. So again, have a look at those lectures slides to have a look at that. We talked about this idea of common or distinct genetic mutations within cells, and what we tend to see is that there's a handful of common of likely suspects, if you like. So it's quite likely if you have colon cancer that you'll have a mutation in the APC gene, because we know that from familiar studies that it's something that's quite commonly observed. You may also have mutations in a tyrosine kinase, some other protein that we don't know about, and a p53. And that might be some of the key mutations that were observed in those cells. Otherwise, you might have a mutation in the APC gene and the RAS or the RAF protein and some other uncharacterized protein leading to tumor. So not everyone has the same mutations in a colon cancer, but there's a bunch of likely candidates which tend to be seen frequently, and each individual biopsy has a mixture of these different mutations. Okay, so there's 35,000 genes in the genome. Maybe there's a couple of hundred that are particularly important for controlling cell growth, and out of those selection of a couple of hundred, there might be 10 or 20 that are more common within a cancer type, and and a particular patient might have that subset of 10 as opposed to that subset of 10. Right. So there's all these sort of likely candidates that occur again and again. And we look at some of the processes and functions of these likely candidates. So that's why we then go ahead and look at receptor tyrosine kinases and RAS and a bunch of signaling pathways, because these likely candidates are interact together within the cell. Okay. There's this concept of another way of thinking about succession of phenotypes or multi-step tumorigenesis, multiple hits within one cell. Another way of thinking about that is looking at clonal selection. So this is the idea that you've got a whole bunch of cells, and one of those cells picks up a mutation, and that particular cell over a period of time is able to outcompete and outgrow the ones around it. So you start to have a higher proportion of these, okay? And then within one of these cells, another mutation occurs, so that over time, this one now can grow at a faster rate than those around it, all right? And then, so you tend to get a new population which is outcompeting the initial population. So the idea is, if you think about Darwinianism and evolution, it was this idea of survival of the fittest, that some species picks up a mutation that gives it a phenotypical characteristic that allows it to survive in a situation in which the others can't. What's a really silly example? Maybe a mutation that gives a giraffe a slightly longer neck. Some sort of point mutation that leads to a growth characteristic or a developmental change. So you've got animals with a slightly different characteristic. If at some point there's a drought and only those top level of trees, those leaves at the very top, the animals that can eat those ones are going to survive, and the shorter ones are going to die off, and therefore you get a shift in the population of the genetic pool, giving rise to animals with these different characteristics. So droughts wipe out populations of organisms, and those that have got a ability to survive the drought through some sort of mutation, they then tend to become the dominant population. So there's all these. I don't want to get into Darwinian evolution, I'm not an expert on it, but that idea of survival of the fittest, that those models of thinking have been applied to thinking of survival of cells and having characteristics that allow you to outcompete the surrounding cells. So it's kind of nice to take this, because evolution is driven by mutations, so it makes sense to try and think about mutations in that context. This idea of linear clonal selection is a fairly simplified, model. It's not the best model. You can then start to consider multiple selections of mutations existing simultaneously, giving rise to slight variants of cells, so that there is some heterogeneity within that population. So initially, when we're thinking about clonal selection, we're talking about a group of cells picking up mutations, and it was like the same cell type. Whereas in actuality, there's a range of different mutations accumulating within that population. But typically, the mutations that are occurring hark back to this idea of coming from a common subset of genes. Okay, and then I guess there's then a bit of a breakdown of some of the common players that are common to colon cancer. Okay, so biopsies from one patient may have RAS and P53 and PRB, whereas biopsies from another patient may have ABC and P16 and one of the SMAD genes. Okay, but these are the common players that are involved in driving growth or slowing growth, and mutations in those are the ones that are important for developing cancer. We then went on to talk about metastasis and how other phenotypes, not just growth, but other sort of taking over other cellular processes are important in cancer as well. Obviously, growth is the easiest one to visualize without an understanding of a lot of cell biology. But as you know more about cell biology, you can start to appreciate how mutations in pathways relating to apoptosis are important, or mutations in pathways relating to growth factors that might encourage blood vessels to grow towards the cells. So there's various factors that can be released by these cells, which manipulate the environment to its advantage. Okay, so then we talked about how in some cells you may have mutations in cadherins, or you might have the release of peptidases that can cleave these cadherins, or that can cleave the extracellular matrix. So all these properties of cells are slowly changing to allow these cells to exist in isolation. Okay, so they're no longer, the growth is no longer inhibited by the neighboring cells, and they can exist in isolation. So again, I don't go into this in any great detail, but if you go to those lectures, you'll see there's a discussion of some of those properties. And then we also kind of link this through to metastasis. So angiogenesis and metastasis are kind of related, and once the cell has picked up these load of mutations, it can then start to manipulate growth through the extracellular matrix, escape into a blood vessel or a lymph system, and then leave the system and invade neighboring tissues. Okay, and some tissues are more susceptible to invading cancer cells than others, and again we talk about some of those concepts. So just to, have a look at some of those slides and some of those concepts. All right, so angiogenesis and metastasis are kind of related. All right. 35 minutes, Jesus. Okay, so we then went on to lecture 2, where we talked predominantly about cellular oncogenes. Okay, so we've talked about in the first lecture about some of the characteristics we observe at a phenotypic level. So this idea of succession of phenotypes, multi-hits, and clonal selection, and some of those concepts. Now we're starting to look at what are some of the actual genes that are modified in cells that give rise to cancer. So the most so when people first started looking at cancers, there was an early theory that it was viruses that were causing cancer, because in the 60s, 70s they were identifying or associating viruses with cancers, and looking at the genomes of the viruses and identifying some proteins. So these were called oncoproteins. But basically the take-home message is that most cancers are caused and driven by mutations in the human genome. Okay, so but from this early work looking at some of the proteins, some of the human proteins that were picked up by the viruses, they led to the discovery of these sequences in the human genome, and so we'll look at some of those. Clearly there are some cancers that are caused by viruses, and we talk about cervical cancer and some others. Okay, but it's unusual for cancers to be causing these viruses to be causing these cancers, and typically even in these viral cancers, it's a viral protein that's interacting with a key tumor suppressor and inhibiting that tumor suppressor. Okay, so what are we doing here? So it turns out, if we just skip ahead here, that these viruses were integrating into the human genome. As part of their life cycle, they were then excising themselves from the human genome and on occasion picking up a bit of human DNA in their genome. Okay, and then it was these bits of human DNA that contained an unlikely event, but over evolution is going to happen, picked up an oncoprotein, and a mutated oncoprotein was then had developed, and then that when the virus was infecting the host, those oncoproteins were being expressed and causing cancer. Okay, so so that was how these human proteins got into the viruses. Okay, and the study of the viruses helped us understand and identify these human proteins. There was a really good experiment here, which is shown here, where they showed unequivocally that it was mutations to human DNA that were driving cancer. So they had some fibroblast cells. So fibroblast cells are cells which grow in culture but that don't contain mutations. Okay, so a lot of tissue culture cell lines are actually mutated cells because they grow faster and more robustly in culture. But at some point in development of techniques, they were finally able to grow normal human fibroblast cells or fibroblast cells from some model organism on a petri dish. Okay, and then they could chemically modify with a mutagen the DNA in those cells, and then extract the DNA from those cells. And then when you then they developed this technique, which again, go to the lectures and we talk about that technique. They developed this technique, calcium phosphate precipitation, to transfer the DNA into another cell line, okay, another normal cell line. Now, the mutations caused by the mutagen then are able to be expressed in those cells and lead to a transformation, like a group of cells that are growing at a faster rate than those around them, which is the first step towards cancer. It's one of the hallmarks of cancer. And even when you transfer those cells back into a model organism, those cells are then growing at a high rate. So within this sort of experimental procedure, it's virus-free. There's no viruses involved. They're taking normal fibroblast cells, they're mutating them with a mutagen, they're extracting the DNA, and then they've got the technique to transfer that DNA into a normal cell line, okay, a normal cell line that hasn't been mutated, and that if you can express that mutated DNA within that normal cell line, that then is giving rise to cells with growth characteristics that are different to those around it. It's growing faster, and therefore it's able to be tumorigenic. So this is really sort of highlighting that human cancers are driven by mutations in human genes. Okay, and we looked in the first lecture about carcinogens or smoking causing mutations leading to lung cancer. Well, in this experiment in mice, they're showing that a chemical mutagen is mutating the genomic DNA, and then if you can transfer that DNA into another cell type, that's causing the mutation. In the same way that the virus was infecting human cells and carrying that mutated DNA and driving cancer. Okay, but the important thing is it's the human genes that are getting mutated in most of the cancers. So as we live our lives, we either through normal processes or through exposure to mutagens are picking up mutations in our cells, and that these mutations on occasion lead to full-blown expression of a tumor. Okay, so so fairly early on it was established that it was these oncogenes that were really one of the driving forces of tumor formation. Okay. We then looked at one of these genes that had been identified as being an oncoprotein, and RAS was one of those. It's such an important gene that it was one of the first ones identified because it tends to be there more often than not in cancer cells. Okay, so they were able to sequence the alleles and identify mutation in a single codon, leading to a different amino acid, leading to a protein with a slightly different property. Okay, and that mutation happened to be in the domain or region of the RAS gene that was its GTPase function, and therefore it wasn't able to hydrolyze GTP to GDP. Now, why is that important? Okay, so let me skip some of this material whilst it's fresh in my mind, and let's jump to this one here. So why is a mutation in a GTPase important for an oncoprotein? So you've got RAS, it's in the off state. It's bound to a GDP molecule. Okay, during normal cellular growth, a process occurs where a guanine exchange factor is able to displace the GDP and allow GTP, which is present at a higher concentration in the cells, to then occupy that space where the GDP was. So we now have the guanine exchange factor dislodging and causing exchange of the GDP, leaving an empty pocket where the GTP can then populate. Okay, so this causes part of the molecule then to have a different structure. So there's a switching loop which is now exposed. All right. Now, that can then activate other proteins that bind to and recognize the new structure, such as a RAF. Okay, but the important thing here is we're looking at how does that mutation, single point mutation in the oncoprotein, lead to its hyperactivation and therefore more growth. Okay, so basically there's a process where GDP is hydrolyzed back to GDP. Okay, and the enzyme responsible for hydrolysis is RAS because it's a GTPase. Okay, it's a weak GTPase, but it's a GTPase. So RAS is able to hydrolyze GTP back to GDP. All right, so this gets hydrolyzed. Okay, which then turns off the signaling. But if the mutation in the domain here, there's a mutation which means the protein is no longer able to hydrolyze the GTP, then it stays in the active state with that loop exposed. Okay, because it can't hydrolyze the GTP. The GTP is causing that to stick out, and RAS was a GTPase will hydrolyze GTP, and that domain pops back in again. That loop can come back down again. But because it's got a mutation, then it stays in the on state, and it can still activate by binding to a downstream effector. So at some point, without the mutation, a normal RAS is a GTPase. It hydrolyzed GTP to cleave off one of those phosphates. The gamma phosphate from GTP is cleaved off, and RAS returns to its GDP bound state, which is off. Okay, so if you remember, I can't remember my structure, but you've got a base here, such as an adenine or a guanine or whatever, but in this case it's GTP, so it's guanosine, bound to a ribo sugar here, and then on the 5 prime, 1, 2, 3, 4, 5, the 5 prime of the ribo sugar, it has 3 phosphates, your alpha, beta, gamma phosphates, and RAS is a GTPase, and it's able to cleave off the gamma phosphate, which convert GTP, the triphosphate, to GDP, the diphosphate. Okay, so it's a pretty basic reaction. Excuse me, that's a bit of biochemistry from second year, but just in case you're not sure what's going on here in the activation of RAS, RAS binds GTP to be activated, and then RAS hydrolyzes GTP to switch itself off. And mutations in RAS that prevent it hydrolyzing GTP lock RAS into the on state. Okay, and there's a a GAP, a GTPase activated protein, which enhances the GTPase activity of RAS. Okay, so again, go back to the lectures for that. So, so we're talking about point mutations in a gene sequence were identified that we now know are mutations in the GTPase activity of RAS and allow inactivate the ability of RAS to hydrolyze GTP. So these kinds of mutations were identified in lots of cancer types, and hence people went on to actually work out what was going on. Other oncoproteins were identified in Burkitt's lymphoma, where the c-Myc gene becomes expressed inappropriately. And again, the lectures go through that process whereby you've got these genes involved in coding for immunoglobulins. So immunoglobulins recognize antigens in the environment, and there's hundreds of thousands of different antigens that we encounter and have to respond to. So we don't have an individual gene for each antigen. That would just be ridiculous. We'd have a genome that was hundreds of thousands of genes big to allow for that. So we have a process of of immunoglobulin gene shuffling or reshuffling, whatever you want to. So that different shuffling of the genome sequence gives rise to different protein sequences, and therefore different binding sites for the different antigens. Okay, again, I'm not an immunologist, so talk to someone who understands this stuff. But what I want to focus on is that there's a region of the genome which is being cleaved and rejoined as a normal cellular process to produce all these different immunoglobulins. Now, sometimes that cleaving and rejoining goes slightly astray, and it gets cleaved and rejoined to the c-Myc gene by mistake. All right, so you've got this promoter here, which is going to be expressing immunoglobulins, is now expressing the Myc gene. Okay, and inappropriate levels of expression, so high levels of expression of the Myc gene occur, and the timing of expression is wrong as well. So you get like a large time frame of expression and inappropriate levels of expression. So another one of these important oncoproteins was identified in Burkitt's lymphoma. So you have the infection leading to the activation of the immunoglobulin genes, leading to the gene reshuffling, leading to the translocation to another chromosome where the c-Myc gene gets linked to the wrong promoter. Okay, and then we go on to look at some other examples of oncogenes which have been identified as being involved in cellular signaling pathways. And typically the receptor is an oncoprotein because a receptor can be locked into the on position and drive signaling pathways. Okay, so I sort of tried to talk about signaling generally from a sort of a fairly generic point of view, just to say that we have receptors on the cell surface, we have adapter molecules that can bind to activated receptors. We then have some sort of signaling cascades, which the ones I talk about involve phosphorylation events, so these different kinases, leading to activation of transcription factors, which turn on genes, and then that can activate a whole new subset of genes, leading to a large phenotypic change. Okay, so that's just a very kind of general slide. Don't get bogged down in all the detail there. And then before we looked at an individual receptor in any detail, we looked at the Src protein. And the Src protein has a lot of properties that the understanding of Src led to a better understanding of the receptors. Okay, so they discovered the Src protein first. So Src protein, I don't know how you think about Src. It's a protein that's got a tyrosine kinase domain. I'll put a K there for a tyrosine kinase domain. It's got tyrosine residues, which, if you remember, tyrosine is just your bog standard amino acid side chain with an aromatic ring with an OH group on it. Okay, so remember, phenylalanine hasn't got an OH group, and tyrosine has. Okay, so the kinase can add it's not deleting. A kinase can add a phosphate group to the OH group. All right, and that's all a tyrosine kinase is. It's a enzyme activity that phosphorylates a tyrosine residue on another protein. Okay, so getting back to our Sark protein, it has a tyrosine kinase domain. It has tyrosine residues, and it has a domain called an SH2 domain that can bind to phosphorylated tyrosine. So the kinase can phosphorylate itself, and then the SH2 domain can then bind to the phosphorylated tyrosine. So effectively, you've got an enzyme that can auto-regulate itself by the kinase phosphorylating itself, and then the SH2 domain binding to the phosphorylated tyrosine, and that sort of then buries the kinase domain within the body of the enzyme so that it can't phosphorylate other proteins. Okay, but other proteins can come along. This is probably too much information, but other proteins can come along with a stronger binding affinity to the phosphorylated tyrosine and displace the SH2 domain from the phosphorylated tyrosine and activate the kinase domain. Anyway, let's not get too into Sark, but an understanding of Sark then led to a way of thinking about receptors. Okay, so I think in this lecture or another lecture, it shows the homology between a receptor tyrosine kinase and the Sark protein. So there's homology there. So it becomes apparent that these receptor molecules have these tyrosine kinase domains. All right, and then that's important to know, because we then go to look at these receptors, and really we should think about these receptors as dimers that are embedded in the plasma membrane. Okay, so this is the plasma membrane, and this is two individual monomers of a tyrosine kinase receptor bound to a ligand. Okay, and then understanding that these are kinase domains, we now understand that the binding of the ligand brings the two dimers together and then allows these kinases to become active, whereas this kinase now is in the vicinity to phosphorylate this cytoplasmic tail, and this kinase can phosphorylate this one. So you get regions on the cytoplasmic side of the receptor that can be phosphorylated on the tyrosine residues by the tyrosine kinase domain. Okay, and our ability to look at receptors and understand how receptors work, a lot of that was informed by understanding how the Sark protein worked. Okay, so Sark was the first oncoprotein studied at any great level and understood at a mechanistic level as a little molecular machine, and an understanding of Sark led to an understanding of how these other molecular machines called receptor tyrosine kinases function. Okay, and it's a dynamic process. You've got individual monomers which are brought together by a ligand, all right, and then that bringing together of these individual monomers then bring together on the cytoplasmic side here these tyrosine kinase domains which allow for phosphorylation of each other. So it's a trans autophosphorylation or cross phosphorylation. All right, so we start to look at how some of these oncoproteins function at a molecular level. So understanding that RAS is activated on and off through binding to these guanosine nucleotides, and understanding that these receptors are activated by binding to ligands, bringing together the tyrosine kinase domains. Okay, and then the lectures also have a bit more of that molecular detail on the activation of RAS. All right, so then we moved on to another lecture. Okay, so now we're moving on to the third lecture, where we're looking at tumor suppressor proteins. Okay, and again, the narrative of the textbook is that we've looked at cancer as a broad phenotype and looking at the development of cancer over time and multiple mutations over time, knowing that multiple genes are involved in the process. Okay, we then looked at a bit more detail as they discovered things in the 70s. We looked at some of the genes whose gene sequences were mutated, and looked at some of the proteins encoded by those and how the mutations affected the function of the proteins. Okay, I mean it's kind of pointless looking at a mutation in a gene sequence unless you understand how that mutation affects the protein activity. Okay, you can't understand how a mutation has an effect. You know it's associated or causative, but you don't know how it actually functions unless you look at the protein and the change in the protein structure. Okay, and also when you're designing drugs for typically the protein activity is predominantly the target of drug design. Anyway, so we're now looking at some research, I guess it's in the 70s, where a researcher who was studying a disease of the eye that occurs in newborn infants, he looked at some of the genetics of this disease called retinoblastoma, and he identified the protein involved in the disease, and it became the first identified tumor suppressor protein in human cells. Okay, and once retinoblastoma was established as a tumor suppressor, the concept of tumor suppressors was applied to other proteins that were being studied as well. But retinoblastoma was the first identified tumor suppressor. Okay, so the narrative then is that we have a bit of an understanding of tumor viruses led to the discovery of oncoproteins, and then we studied the oncoproteins in the human genome and looked at some of the functions of these oncoproteins. Now, a mutation in a mutation in a human oncoprotein is a dominant mutation. So we've got two alleles of each gene, as you know, and a point mutation in one allele leads to a dominant mutation. So even though the cell's still producing a normal proto-oncoprotein, normal gene product from one allele, the other allele is producing a mutated hyperactive protein which dominates, and the good copy can't protect the cell from the overactive copy. So if it's something that's driving growth, then the one that's more active dominates, and the one that's less active is not inhibiting the activity of the other one. It's a pretty simple concept. I'm over explaining it to you. Okay, so in that sense, it was thought that the properties of these mutations were dominant. In other words, that the cancer phenotype was dominant over the normal cellular phenotype. So if you had mutations in these oncoproteins, it was the dominant phenotype. Okay, so there's this kind of cool experiment that was done that involved cell fusion. So if you take if you take a cancer cell and a normal cell, assuming that the cancer cell has a dominant phenotype, when you merge the two cells, then the active thing from the dominant cell will dominate the fused cell. All right. But what turned out is that what happened is that when you fuse one of these cancer cells with a normal cell, the phenotype that resulted was that the cancer phenotype was recessive. Okay, and that shocked a lot of people, I think. So it turned out there had to be something else that was more common in cancers that was causing the cancers, and this led to the development of these the idea of looking for tumor suppressors. And if you remember in the first lecture, we stated that tumor suppressors are more common in cancer cells than oncoproteins. Okay, that using this experiment, they were able to demonstrate that some of the controlling factors that lead to cancer formation are recessive. Right? So effectively, the good copy of the tumor suppressor in the wild-type cell was able to restore the function that was lost in the cancer cell. Okay, so the occurrence of these tumor suppressors has been flagged as being an important thing to understand, and the oncoproteins were the exception to the rule. Okay, so Alfred Knutsson, this I think he was Swedish, Scandinavian scientist, he was studying the occurrence of retinoblastoma, and he was looking at the genetics of the occurrence of retinoblastoma, and he'd identified that you have cancer in one eye or both eyes, so it can be unilateral or bilateral, and he was able to conclude that bilateral retinoblastoma was an inherited form of the disease, and effectively during development both alleles were affected, and that led to the development of the disease in both eyes, whereas the sporadic form of retinoblastoma, which occurred after fertilization, would occur in the group of cells that became one of the eyes, not in the group of cells that became both of the eyes. Does that make sense? So later on in development, the mutations must have occurred. Okay, so it occurred in the cells within this region of the body, as opposed to a mutation that had occurred earlier in development that gave rise to both eyes. Okay, so they were able to look at the occurrence of the mutations, and to cut a long story short, the question was, how do you knock out both copies of these alleles to give rise to the disease in a short period of time? So from the point of fertilization to embryo development was a 9-month period, and you were having to have 2 independent mutations to knock out both copies in the sporadic form of the disease, which was highly unlikely, but yet there was still a proportion of children who were born with the sporadic form. And also, if you look at the familiar inherited form, then you had to have another mutation in the good copy during that early stage of development to give rise to bilateral retinoblastoma. So the case against tumor suppressor proteins was the unlikely occurrence of mutations in both alleles, because each mutation has a low occurrence rate, and therefore to get both alleles knocked out was highly unlikely. But it was seen, so something was going on. So he came up with these processes which led to the loss of heterozygosity. So you've got one mutant allele and one good allele, so it's heterozygous, okay? But for the full-blown disease, it has to be homozygous, because the good copy can protect against the bad copy. So you have to have a mutation in the good copy. in addition to the existing mutation. So effectively what he was saying is that through, and I won't go into the details here, you need to go to the lectures to understand this, but through the process of mitotic recombination, you can give rise to offspring with homozygous mutations in both copies, okay, rather than heterous outcomes. So effectively, like I said, you need to go to the lectures to look at that. And also there were other processes involved in losing the existing good copy, which involved gene conversion. Okay, so the DNA polymerase was jumping between, is thought to jump between the two alleles and then copy the mutant copy into during DNA replication. All right, so we discussed the unlikelihood of tumor suppressors needing to knock out both copies, and we talk about other cellular processes which are more frequent than mutations, which can lead to one mutation being copied, giving a higher frequency of occurrence. So, and then this led to the discovery of the Retinoblastoma gene or protein, and in terms of the genetics of this, it differs from that of the oncoproteins, whereas oncoproteins, you have a hyperactivation of one allele, and it's a dominant mutation, whereas in the tumor suppressors, you have to knock out both copies. So you're not enhancing the function of the retinoblastoma, you're actually inactivating the function of retinoblastoma, but because you've still got a good copy, you have to inactivate both of them. Otherwise, the good copy can protect. And if you think back to this experiment here, it clearly showed that when you fuse two cells, a good copy from the normal cell can protect against the loss of function in the cancer cell. So same kind of story here. The good copy can protect against the mutated copy, and therefore you've got to knock out both copies. All right. We then look at the actual function of retinoblastoma in cells, and because it's one thing to know that you've got to knock out both alleles to give rise to disease, but that tells you nothing about the cellular process that's involved. So it was important to study the retinoblastoma protein and look at how it functions, and that tells us an awful lot about the molecular biology of normal cells, because if you understand how a normal cell works, then when that process breaks, it leads to the cancer or the inappropriate growth. So let me sort of skip down here to talk about the function of retinoblastoma first. So we go right to the back here. We've got these slides here where we talk about the function of retinoblastoma. Now, retinoblastoma is a phosphoprotein, so it's phosphorylated during normal cellular growth. Early on, the retinoblastoma is hypophosphorylated, meaning it doesn't contain many phosphate groups. Okay, so the serines and threonines are not highly phosphorylated. During the hypophase, it can bind to a transcription factor called E2F transcription factor. Okay, and that stops E2F from activating genes through gene expression. At some point during cell growth, retinoblastoma becomes highly phosphorylated. So it becomes phosphorylated. When it's phosphorylated, it can no longer bind to the E2F transcription factor, and therefore it allows the expression of genes. Okay, so just to really quickly summarize the function of retinoblastoma, it suppresses the function of a transcription factor. Okay, when retinoblastoma is a phosphoprotein, and when it's highly phosphorylated, it lets go of the transcription factor, which allows gene expression. Okay, there's more to it than that, but at a rudimentary level, that's what you need to know before you can start to understand how it functions. Okay, so we'll come back to the role of retinoblastoma later. So another thing to look at is how these tumor suppressors can be inactivated through their promoters becoming inactive. So effectively, the gene sequence of the tumor suppressor is good, but it's simply not being expressed in cancer cells. Okay. Now, one of the ways that one of the things that's been observed in cancer cells is that the promoter region of tumor suppressors are highly methylated. Okay. So how does highly methylated promoter region lead to gene suppression? All right. So what you need to understand first is how generally the process of gene suppression operates, and then we'll discuss how methylation affects that process. Okay? So gene suppression as a general process is controlled at the level of nucleosomes and at the level of histone acetylation and deacetylation of lysine residues of histones. Okay, that hopefully you have an understanding of this. So firstly, what is a nucleosome? Okay, so a nucleosome is an octamer of 8 histone proteins. Okay, they just form this thing, this lump. Okay, and around that octamer of histone residues is 2 wraps of DNA. All right. Now, the histone residues have tails that protrude, and these tails are rich in lysine residues. Okay, and lysine residues, as you know, are positively charged. Okay. Now, positively charged protein and negatively charged sugar phosphate backbone of DNA interact and cause tight wrapping of DNA around the nucleosome. Tightly wrapped DNA cannot be expressed. Okay, so the cell has a process of these enzymes called histone acetyl transferases. Okay, and what it does, it at the point where the lysine residues are positively charged, it adds an acetyl group, which that process of modifying the amino acid residue, it loses its charge. So these lysines become acetylated, but more importantly, they lose their charge. So all of these positive groups are lost and acetyl groups are added. Okay? Acetyl groups are these small chemical, small little things. Okay? So the positive charges on the histone tails are lost. So the nucleosome becomes less positive. Therefore, the nucleosome proteins interact less with the negatively charged DNA. So the DNA becomes loosely compacted, so that the DNA now can be bound by transcription factors, because the DNA is not tightly bound to the histones. All right? So we have this process of acetylation of histones, which loosens up DNA by removing the positive charges. Okay? So if you've got DNA that's tightly wrapped around a nucleosome, it's inactive. All right? If the DNA is methylated highly, such as in cancer cells, then the methyl groups, they attract something called a HDAC. All right? So just think of these other proteins, and they're histone deacetylases. So the histones, they then take the acetyl groups that were attached to the lysines, and they deacetylate and return the positive charge. So the methyl groups brings in the histone deacetylase, which removes the acetyl groups. It deacetylates the histones and returns the positive charges. And that tightens up the nucleosome, and a tight nucleosome can't be expressed. And if this happens at the promoter of a tumor suppressor, then the tumor suppressor is turned off, and therefore growth is driven, allowed to proceed when it shouldn't have proceeded, which is what happens in cancers. So you've got this process then, highly methylated promoters in cancers bring in histone deacetylase, which takes the acetyl groups off the histone tails, allowing the positive charge to return, allowing the tightening of the nucleosome, switching off the gene. Okay? And it's important to understand how a process works, otherwise you'll never understand how cancers form and how drugs are developed to manipulate cellular processes. Okay? So it might sound a bit complicated, but if you think about it and read about it and look at the lectures, it becomes quite straightforward. Okay? Now, another thing that's interesting about this process, okay, so we've got a process of gene suppression through methylation. Another thing that's really interesting is that when they looked at retinoblastoma in more detail, I don't know if I've got a slide about this. Yes, here. So when they looked at about this process of how does retinoblastoma function, it turns out that retinoblastoma can tether a histone deacetylase to affect gene expression, and that the E2F protein that retinoblastoma is also controlling, when in the absence of retinoblastoma, the histone, the E2F can bind a histone acetyl transferase. So effectively, retinoblastoma is not only binding to the transcription factor to control gene expression, but retinoblastoma and that transcription factor are also manipulating the structure of DNA around the nucleosomes through controlling acetylation. Okay, and methylation affects that process, and retinoblastoma and the transcription factor also affect that process. Okay, so again, I won't try and describe that to you now. It's in the lectures. I forget how much detail I described it in the lectures, but it's a fascinating bit of biology, and it's really interesting to see how DNA structure affects gene expression as a fundamental process, and it's interesting to see how in cancer cells that process has gone wrong through inappropriate levels of methylation. Okay, so so the next thing we went on to look at in the lecture, and it's related to what we've just been talking about, but the other thing we went on to talk about or look at then was here. It was the function of retinoblastoma, and we've talked a bit about that, but also this thing called the cell cycle clock. Okay, so effectively, if you've got a cell, it doesn't just keep dividing continuously. Okay, it is told when to divide. Okay, and that's a process that's really well understood. Okay, and effectively, a growth factor can drive this process. Okay, but how? That's the question. How does that happen? Okay, so effectively, the growth factor leads to the activation of a signaling pathway through a receptor, which leads to gene expression. Okay, and we'll talk about that later. Now, the growth factor leads to gene expression is the key part of that sentence. And one of the genes that's being expressed is a thing called a cyclin. All right? So we're going to look at how these cyclins are involved in controlling the cell cycle. Okay? Retinoblastoma also has a role in this, so it's good that we've got an understanding of a bit about what retinoblastoma does. But now, important message is growth factors can drive signaling pathways to lead to gene expression, and one of the genes that can be expressed is a cyclin gene. All right? So we're talking about signaling and about how receptors, remember our tyrosine kinase receptors on the membrane of the protein of the cell, they can bind to a signaling molecule, and that can activate a kinase domain to drive a process leading to gene expression. Okay, so now clearly cells aren't just growing all the time, because that's the hallmark of cancer. So cells are effectively not growing unless they're told to. So this process of dividing is not occurring, all right, until the growth factor is present, and the growth factor then causes the growth to occur, all right. So this little diagram here, I'm looking at how the growth factors can lead to activation of growth. Okay, if you're thinking about cell growth, there's two ways of thinking about growth. There's replicative growth, and there is just growth getting bigger, something getting bigger. Okay, when we talk about cell growth, we don't talk about a cell getting bigger. I mean, it changes size a bit, but we talk about replicative growth, cell doubling. Okay, so if you're going through a cycle of cell growth, then we have the cell cycle, which goes from G0, which is not growing, into G1, which is a gap phase between where the cell is building up resources, but it's not actually dividing yet. And then we go into S phase, where the DNA replication occurs. And then we go through another gap phase called G2. And then we go through mitosis, where the cell division occurs. Okay, so you need to replicate the DNA. So you've got 2 copies of everything before you can then segregate that DNA into 2 cells to divide them. Okay, so you've got 2 gaps, and then you've got 2 important processes of DNA synthesis and mitosis, and then you've got a quiescent phase where the cells are sitting there, but they're not growing, but they're metabolizing and doing useful things. Okay, so to understand uncontrolled cell growth, you need to understand that there's phases of the cell cycle leading to cell division, and because cells aren't growing, then they're obviously paused at some point in this cycle. So this cycle takes time, a couple of hours to go through, but typically the cells aren't growing, they're just sitting there metabolizing and doing useful things. So during this cell cycle then, there's a pause. So a point, a particularly important pause, is called the restriction point, the R point. And that occurs late in G1, just before DNA synthesis. Okay, so there's a bunch of these checkpoints. The one I particularly want to talk about is the restriction point that occurs late in G1. And that's where retinoblastoma has a role to play by inactivating the E2F transcription factor. And also during this G1 phase, this is when cells are receptive to growth factors, okay, leading to cell signaling, leading to gene expression, leading to expression of a cyclin, amongst many other things. All right, so if you look at cells in a continuous culture, or if you are able to synchronize cells some way, and you looked at these phases of G1, S, G2, M, G1, S, G2, M. If you look at cells going through that process, then you can take cell aliquots at various points in time, and you can look at gene expression at various points in time, and you can look at what's expressed. And they did that, and they noticed that these cyclins are peaks in expression of certain proteins. So these are cell cycle regulated proteins. Proteins whose expression is determined by which stage in the cell cycle it's there. Okay, so lots of proteins are expressed all the time. Some proteins are only expressed when they're needed, and some genes are responsive and they respond to the environment in some way. But these ones are responding to factors which are involved in cell growth. Okay, so the cyclin gene is one of the genes we want to talk about. So we have this diagram here. We know some of these proteins are cell cycle regulated, and what this little diagram here is showing is that the growth factor is working through a receptor tyrosine kinase, leading to activation of RAS, leading to the MAP kinase pathway, leading to gene expression of a cyclin. and that occurs during G1. Okay, so what's happening here is a growth factor is leading to activation of a receptor, cell signaling, and expression of a gene, or lots of genes, but some of them being cyclins. Okay. So why is it important that we have the expression of a cyclin? Well, the cyclin protein, it forms a dimer or a heterodimer with a cyclin-dependent kinase. Okay. Now, this occurs in late G1 because that's when the cell's responsive to growth factors, and one of the proteins, there's lots of them, but one of the proteins that these kinases can phosphorylate is the retinoblastoma protein. Okay, so we talk about then the phosphorylation of the retinoblastoma going from hypo, less lightly phosphorylated, to hyperphosphorylated. Okay, due to these cyclin-dependent kinases, which are activated by growth signals. Okay, and what's the outcome of this? Well, if we think of retinoblastoma as this kind of shaped thing, it's bound to E to F. So this is RB, and that's E to F. When you phosphorylate the retinoblastoma, it can release the E to F, or at least that's a simple way to think about it. And then the E to F can then bind to a promoter region of a gene to drive transcription. All right, and if you think about it, you've got a growth factor in G1 phase driving expression of genes. So what sort of genes would be required to enter DNA replication, which is the S phase of the growth cycle? Well, it would be the genes involved in DNA replication. Okay, so your polymerases, your genes that make nucleotides, which are required for polymerization into a new DNA molecule, and all of those factors that are required for DNA replication are turned on. If you look at those genes, you'll see that a lot of them have binding sites for the E2F transcription factor. So the growth factor causes activation of the cyclin-dependent kinases, which then phosphorylate the retinoblastoma, which release the transcription factor to turn on genes required for DNA replication. All right, so again, there's more to it than that, because we also know that retinoblastoma and E2F can bring in histone acetyltransferases and histone deacetylases, and we talked about nucleosome structure as also playing a role in gene silencing and gene expression. But at this level here, we're just talking about looking at the role of retinoblastoma in terms of how it binds to the E2F transcription factor. All right, so that was it for that lecture. Let's move on to the next one. Okay, so let's now move on to lecture 4, where we look at some of these signaling pathways in a bit more detail to understand how a growth factor is able to activate gene expression. Okay, and we're concerned in this lecture with understanding what a signaling pathway is. What does it mean that we have a signaling pathway? What is a signal? I mean, if I was talking about an electrical circuit, then it's quite easy to visualize how the electrical signal passes through, all the resistors and capacitors and diodes, and how you can control the flow of electricity from one side to another side of a circuit board, and you can track it really well. And you can get your soldering iron out, and you can start to replace bits and swap things around and invert things, and really break it down and understand how that works. Okay, and you can then sort of look at signaling and wiring of entire cities, about how telephone networks is connected and how the copper cables are connected from house to house, and really understand how things are connected and how signals pass between things. And the goal of biology, molecular biology, is to understand how signaling works within a cellular environment. So what does it mean to what actually is a signal, and how does it pass from one region of the cell to another region of the cell, and how does it activate something to occur? So in the last lecture, we looked at how gene activation can occur. We talked about how tightly wrapped DNA around nucleosomes can be modified, and about how transcription factors can come in and bind, and we're starting to think about processes at a cellular level. And in this lecture, we're going to look at a signaling pathway at a cellular level, or a mechanistic level, within some level of detail. Okay. Again, it's very simple, but it's starting to think about things in molecular terms. So, in the slides here, I'm showing this notion that if this is the plasma membrane of the cell, and this is the nucleus of a cell, that on the surface there's lots of these receptors, okay? And each receptor is responsive to different factors, and these factors are driving different processes, and you see it all the time on the web and in textbooks, all of these pathways, and it becomes a hairball of interactions and pathways, and it's difficult to visualize and focus on what's going on. So I think it's more productive to look at a cell, the plasma membrane and the nucleus, with the DNA down here, and just look at a linear pathway. So one pathway that starts but with the binding of a ligand to a receptor, and that goes in a fairly succinct way to lead to gene expression through a bunch of things, all right, proteins. And I think understanding one pathway, which is a linear pathway in a rudimentary level, is really important to your thinking and understanding about other pathways later on, as you start to know more about cell biology. Okay, so the expectation is then we can understand how signaling occurs along a pathway, and the pathway that I talk about, which is well understood and the textbook focuses on, is a signaling pathway that starts with a receptor tyrosine kinase. It goes through the RAS protein and leads to the activation of some transcription factor, such as ERK or something like that. All right. So that's the. But what does it mean? What is the actual thing here that constitutes the signal moving from the plasma membrane to the nucleus? Okay. And to understand that, we've got to look at, well, what's this protein, what's that protein, and what's the interaction that occurs between those two proteins. Okay, and then go to the next one along in the sequence and go, well, I know what this protein is, what's this protein, and what's the interaction between those two, and then what's the next one, and then what's the next one. Okay, and it's a bit like molecular Lego. You've got something here that connects, and leads down into the nucleus. All right. So we then started to look at these things. In terms of how you think about a cell, I just flagged the work of someone who I think is really quite impressive, this guy called David Goodsell. And he's a scientist. He works at PDB, and he's involved in curating those three-dimensional structures of proteins on the website there. And he's a research scientist as well. But he's also an artist, and he sort of draws things in the cell in relation to each other. And I think it's worth, if you're interested, to go and have a look at some of his artwork, which is anatomically correct, if you like, in terms of the size of these molecules and the numbers of these molecules in the cell. Anyway, let's focus on one of these linear cell signaling pathways, as opposed to the pea soup that is the reality. Okay, so let's look at one of these linear cell signaling pathways. And what I'm going to tell you is that in signaling pathways, you get a receptor being activated, leading to the activation of a gene, okay, or a bunch of genes. And then this leads to gene expression, transcription, translation to give new proteins. All right. Some of these proteins are also transcription factors, which then go to another region of the DNA to activate a second wave of proteins, and yada yada. This is an ongoing thing. Okay, so this occurs. It was known to occur for a long time, but using some experimental techniques, they showed conclusively that this is happening in response to these ligands, these growth factors. That the growth factors were signaling to turn on sets of genes, which were the first wave of things that were turned on. These early sort of transcripts, and then these newly synthesized proteins, which it might take an hour to make a new protein, these newly synthesized proteins, some of those were then transcription factors giving rise to a second wave of transcription, which occurred an hour later. Okay, so there's a clear timeline of this. So before we got into signaling, looking at an actual signaling pathway, we looked at some of the evidence using microarray techniques that just showed that a bunch of genes were turned on immediately early after signaling, and another bunch of genes was turned on later after signaling. Okay, and without going into the details of those experiments too much, they identified this process of signaling leading to gene activation, which gave rise to new proteins, which then led to more gene activation. Okay, so that came out of fairly recent studies looking at microarrays and gene expression profiles. Going back to the narrative in the textbook, they wanted to understand how a signaling process works. What is a signaling process? So they knew that that RAS was important, and we talked about the RAS activation cycle and how RAS is switched on and off, but they didn't know what was leading to activation of RAS. Okay, then somewhere along the line they discovered the Src protein, and then once they understood that the Src protein was a tyrosine kinase, they then discovered these receptor molecules which also had tyrosine kinases domains in them, and we've talked about that. Okay, so they knew that the tyrosine kinases were oncogenes and they were important in signaling. They knew RAS was an oncoprotein, it was important in signaling, and mutations and hyperactivation of these proteins were seen together. So clearly there's a pathway there, but they didn't know what it was. So again, I'm not going to go into the level of detail that I go to in the lectures, talking about, why SOS is called SOS, and the Drosophila research that led to the discovery of SOS, and the yeast research that led to the discovery of, the guanine exchange factors and all that kind of stuff. But I'll just sort of describe to you a linear signaling pathway. So what we know here is that the ligand, the growth factor, binds to these monomeric receptor molecules and brings two of these proteins together to form a dimer. It can be a heterodimer or a homodimer depending on the cell type and the proteins involved. So we're looking generically here at receptor tyrosine kinases rather than an actual protein. There isn't a protein that's called receptor tyrosine kinase. You've got various receptors. Okay, but in this generic version here, we've got two monomers of this thing that we're going to refer to as a receptor tyrosine kinase. The ligand brings them together, and that causes phosphorylation of the receptor. So the first step in the signaling is the binding of the ligand. The second step in the signaling is the dimerization of the receptor. The third step in the signaling is the close proximity leads to the phosphorylation of the tyrosine residue by the tyrosine kinase domain. Okay, so you can slowly build up what is happening. Okay, if you say the ligand activates the receptor, that is not speaking biology. Okay, what does activation mean? Okay, if you know what activation means, then you need to express what activation is, what the signal is. And activation here, like I said, it's the ligand causing dimerization, causing the close proximity of these kinase domains to then have an opportunity to phosphorylate the substrate, which is the other protein, the other part of the receptor. Okay, so effectively you're getting the tyrosine residues on the cytoplasmic side of this protein that spans the membrane. The phosphate groups on the cytoplasmic tails is important. Okay, why is that important? Well, Sark told us about tyrosine kinases. Sark also told us about domains of proteins that bind to phosphorylated tyrosines. Okay, here we have a phosphorylated tyrosine, and what's known is that there's another protein called GRB, I think it's GRB2. It binds to a phosphorylated tyrosine. And how does it bind? It binds because it's got a domain that's homologous to what was identified here. It's got an SH2 domain. And it binds to a phosphorylated tyrosine, happens to be a specific interaction of a particular phosphorylated tyrosine on the cytoplasmic tail. Okay, and the lectures talk about a whole bunch of these likely candidates where things could bind. Somewhere down here, there's a whole bunch of different tyrosines that can be phosphorylated, and each tyrosine can bind a different thing. Okay, so you've got to think about the cell type, the timing, and what's going on here. Anyway, you've got an adapter molecule, a molecule here which is a protein that has no enzymatic function, except it can bind to a phosphorylated tyrosine. It's also got an SH3 domain, which is a binding domain that can bind to a proline-rich region of another protein. Remember, molecular Lego, bringing things close together in proximity is how signaling works. This binds to something which is called the SOS protein. Okay, the SOS protein, when we did our RAS activation loop, remember, with the off and the on, and we had the guanine exchange factor leading to the exchange of the GDP to GTP. Well, SOS is a guanine exchange factor. SOS leads to the exchange of RAS from a GDP to a GTP. Okay. Now, RAS is also bound to the membrane through something I talk about in the lectures. So again, we've got close proximity and of these things on the plasma membrane where all the action's happening here. Okay, but we want to get the action down to the nucleus, which is where gene expression happens. So we get the GTP being exchanged and the activation of RAS exposing its loop. Okay, changing conformation. This is then recognized by downstream effector molecules, such as a protein involved in the MAP kinase pathway. So you get your typical RAF, your is it MEK, and then ERK pathway, which is a series of and again you need to go to the lectures to brush up on this, but these are each of these proteins is a kinase, okay, serine threonine kinase, and the this is generically referred to as a MAP KKK. This is referred to as a MAP KK, and this is your MAP K. Okay, so this is your triple kinase, your kinase. It just means it's the first player in the pathway that can phosphorylate this, and this can phosphorylate that. Okay, And these things are highly conserved over eukaryotes. So in yeast, these things were initially identified, and in humans, they have been identified. So we have this generic scheme here so that we can talk about the specific pathway in different organisms, because the protein names are different in each organism. So we have this MAP KKK nomenclature, so we can talk about a similar pathway in fruit fly or zebrafish or yeast or any organism that you want to talk about that's eukaryotic. Okay. So anyway, this leads to the signaling pathway now has gone from the binding interaction between an adapter and a guanine exchange protein through to the physical exchange of the nucleotide to a GDP, from a GDP to a GTP, and this is how the signal is moving. Okay, and then that leads to the exposure of a region, the switch loop of the RAS protein, and there's in the lectures there's structures of RAS and how the structure changes on binding of a GTP, and then that leads to the binding and the activation of the MAP kinase pathway, leading to the activation of a transcription factor, which then leads to, as we know, transcription, translation, and protein synthesis. Okay. So that's the main signaling pathway that I was talking about in the lectures, and I gave some of the background information about how our understanding of the SOS protein came from Drosophila, and our understanding of different proteins came from different model organisms, leading to our understanding of what happens in human cells. Okay. What else did we talk about? Talked about that. This is getting back to the structure of SARC, which we've already talked about, how SARC has a kinase, tyrosine kinase domain, a SH2 binding domain, and it has some tyrosine residues that can be bound. It also has other domains which are important and occur again and again in different proteins. So different proteins have these different domains which allows them to interact together to form these different pathways. So if anything has an SH2 domain, then it's a protein that can bind to a phosphorylated tyrosine in another protein. And again, these are specific interactions between individual proteins. There's a nice little model here of what an SH2 domain looks like. So part of that domain of the protein, which was identified in the SARC and is also contained in the GRB, it has a region that can bind to a phosphorylated tyrosine, and it has a region that can bind to other amino acids close to the phosphorylated tyrosine. So it's a very specific interaction between two individual proteins. And so if this is the RAS protein here, so if this is the RAS protein here, things that are signaling down to RAS, they're referred to as upstream factors, and things that are signaling down from RAS are referred to as downstream factors. So we've looked at upstream signaling to RAS from signaling pathways. So what does RAS activate? So we've talked about one downstream pathway, which was the MAP K pathway. And then another important pathway that RAS activates was the phosphoinositol 3 kinase pathway, the PI3K pathway. All right. So again, we don't go into too much detail about that pathway, but what we do know, what we describe in the lectures, is that in the inner leaflet of the plasma membrane are these phosphatidylinositol molecules, and these PI molecules have a sugar attached to them, which is an inositol sugar. Okay, so hence phosphatidylinositols. Okay, now these sugars obviously contain lots of OHs. I can't draw properly, but lots of OHs, and OH residues in the same way that OH residues on serines, threonines, and tyrosines in amino acids can be phosphorylated, in proteins can be phosphorylated. So these hydroxy groups can also be phosphorylated by an enzyme. There's a whole bunch of enzymes. The one we talk about briefly is PI3 kinase, which can phosphorylate our OH to OB. You can add a phosphate group there. Okay, and if you look at the lectures, there's different residues on the inositol that can be phosphorylated, and the pattern of phosphorylation leads to a sugar that can be recognized by different proteins. Okay, and there's lots of proteins that can recognize different versions of the inositol depending on where it's been phosphorylated. Okay, there's also another pathway that cleaves the inositol into a free molecule, which is IP, I think it's called IP2 or IP3, I'm not sure. We don't talk about that in this lecture series, but that can go on to regulate levels of calcium in the cell, which can drive signaling molecules and signaling pathways. But we're not focusing on that in this course. But what we do know is that what we focus on in this course is the PIP2 and PIP3, which is the different phosphorylation status of the inositol sugar, and we have the PI3 kinase, which is phosphorylating PIP2 to PIP3, and we have the P10, which is a phosphatase which cleaves off one of the phosphates into PIP2. Okay, now when PIP2 is present in a cell, it can be bound by various proteins, and we look at some examples of that, such as the protein kinase B, or AKT, as is also referred to. And when protein kinase B binds to the inositol ring, it becomes activated, and then it can phosphorylate lots of things. Okay, but if this is not active and this is active, then this is converted to this, and a different set of pathways is active. Okay, in cancers, often this is mutated so that these pathways predominate. So if this is mutated, then PIP3 is locked into PIP3 state. When the conversion happens here, there's no off switch. And if that's mutated, then this exists, and therefore these pathways leading to cell growth and proliferation are driven. Okay, so again, the activation of the PI3 kinase occurs through the RAS protein. So as shown here, RAS can activate the MAP kinase pathway. RAS can also, through its effector domain, it can also bind to PI3K. Active PI3K can then, again, this is happening at the plasma membrane because RAS is bound to the plasma membrane, and the phosphoinositol sugars, I can't draw them properly, they're also at the membrane. So when PI3K becomes activated by RAS, it can then start to phosphorylate these things to drive other processes. Okay, there's a lot more to what RAS can do, and if you're interested in cell signaling and cell biology, then have a look at some of the review papers on RAS. It's really interesting what RAS can do within the cell. And then lastly, in a very rudimentary kind of way, I just mentioned two more types of receptor molecules on the cell surface. So we've talked about receptor tyrosine kinases. There's another class of molecules that are very similar to receptor tyrosine kinases, but they have an associated kinase rather than an intrinsic kinase. So remember, when we're doing our RTKs, I was talking about this intrinsic kinase, which is a part of the molecule. Okay, the domain of that molecule is a kinase domain. These receptors have no kinase domain, but they bind to another kinase. Okay, and these pathways are particularly important in cytokinin signaling, and these receptors are important in those pathways. And the way this pathway is called the JAK-STAT pathway. So if this is the molecule, the JAK is the Janus kinase that's bound to the receptor, and that's what the JAK is. It's a kinase. It's the associated tyrosine kinase, and the STAT is the thing that binds here and gets phosphorylated. Okay. It can also recognize a phosphate group here. So this is phosphorylating here, like in tyrosine kinases, leading to phosphate groups that are recognized by the STAT proteins. Okay, and then the STAT proteins at some point break away, and this STAT protein here, which is phosphorylated, is bound by this STAT here, which is phosphorylated. Okay, so this STAT can bind to that STAT. This STAT binds to that STAT. So these two STATs break away and form a dimer. Okay, and it's really clever biochemistry when you look at the interaction and binding of these molecules and the activation of these molecules through a signaling molecule here. It's a variant on what we see in receptor tyrosine kinases, but these are receptor-associated kinases. So you can think about evolution and the loss of a kinase, but then the interaction of a kinase supplementing that, and then the different sort of signaling pathway evolving from the original pathway. So it's all really fascinating kind of stuff. And another class of signaling molecule, which again we could have a whole lecture on, and I'd love to give lectures on this kinds of stuff, but it's the G protein coupled receptors. So it's important to know they exist because they have such an important role in the cell, but we just don't have time to go into a lot of detail. So these are these G protein coupled receptors. Now these are different because they're not dimers. Okay, it's a single molecule. So it's called a 7-pass transmembrane thing. So it starts here with the ligand binding domain, and it goes 1, 2, 3, 4, 5, 6, 7. Okay, so it passes the membrane 7 times, and it's a G protein coupled receptor. Okay, it doesn't have a G protein. We haven't talked about G protein yet. This is just the receptor that binds the ligand. Okay, now binding the ligand causes a change in conformation of this bit here. Now this receptor is associated with, as the name indicates, it's associated with a G protein. It's a G protein coupled receptor. So here's the G protein. Now the G protein is part of a trimeric complex. So this is the alpha subunit, beta subunit, and the gamma subunit. All right. Now this alpha subunit is the G protein. Okay. Now, where have you heard the term G protein before? We've talked about RAS. RAS is a GTPase. That's what we mean by G protein. It's a GTPase. We know RAS can bind GDP, and through a guanine exchange factor, it can be nucleotide exchange. It will bind to GTP through a process we've talked about. All right. So you're familiar with GTPases. This is a GTPase. Okay, this alpha subunit is a GTPase. The receptor itself, this domain here, acts as a guanine exchange factor to exchange GDP in the alpha subunit to a GTP in the alpha subunit. Okay, it's very similar to what we see in RAS activation, but this is a different kind of receptor, which is the 7-pass transmembrane receptor. It binds a ligand. It activates causes a change in conformation of the protein here, the cytoplasmic tail, which can act as a guanine exchange factor to activate the alpha subunit. And we don't go into too much detail, but the alpha subunit here can break away from the beta and gamma subunits when it's bound to GTP, because we had the signal, and the activation of that has caused the exchange here. Okay, this can then activate other things in the cell, and just to finish off, it can activate an adenyl cyclase protein, and then that can then convert ATP to cyclic AMP, which can then activate protein kinases, can then go away and phosphorylate things. And as soon as you've got activation of kinases, you can envisage that as being signaling molecules leading to activation of gene expression. Okay, so these are other sort of signaling pathways that we don't have time to get involved with, but I'm sure in some of the projects you've been doing, you've been coming across mutations or tumor suppressors and oncogenes that are involved in pathways driven by JAK-STAT pathways, or that are driven by G protein coupled receptors. Okay, so the world doesn't revolve around receptor tyrosine kinases, but that's the linear signaling pathway that we've focused on in the lectures, but just be aware that there are other signaling pathways that are important as well. All right, that wraps it up for that lecture. Okay, so this brings us to the last lecture of this series, which is the rational treatment of cancer. So the idea was here that we've discussed aspects of cell biology that give us a better understanding of some of the pathways and some of the key proteins that are involved in controlling cell growth, and also some of the key players that get mutated in cancers. Okay, so the idea is now, does this information, can it be used to as a flow-on effect to think about a rational treatment of cancer, so rational drug design. Okay, so what do we mean by rational drug design? Well, when you think of some of the more popular anti-cancer drugs that exist currently, they've been around since the late 60s, early 70s. So they came into being before we knew about oncoproteins and tumor suppressors and all of the other things about cell biology. So these older drugs weren't rationally designed. I think the term is serendipitous design. It was designed by chance. It came across somehow. So one of the examples here, as shown here, is the compound, one of the ones I talked about, a serendipitous discovery, was cisplatin. And this character, Barnett Rosenberg, was studying the effect of electric currents on the elongation of bacteria. So I'm not quite sure what he was doing, but the way I think about it is that we all know that we have these electrophoresis tanks, okay, and they've got positive and negative electrodes on them, and we put our DNA gel there, and we fill them up with buffer, and the electrodes are attached here to these platinum electrodes, okay, and the platinum because they're inert. And in this buffer here, you've got various salts, chlorides and ammonia compounds, and tris buffers and all that kind of stuff. So we're used to this sort of thing. Now, he was somehow looking at the effect of passing electric current through, effectively through bacteria. So he had a system where he was running growing bacteria in a liquid and running an electric current through there, and there was an interaction between the platinum electrode, which is thought to be inert, go figure, and the buffer, and that was leading to the production of cisplatin. So platinum with 2 chlorides and NH3s. So this compound, sort of cisplatin, was being produced unbeknownst to Barnett Rosenberg, as he was studying the effect of the electric current running through the buffer. Turned out the elongation of the bacteria was caused by the cisplatin molecule, and he figured, well, if it stops bacteria from replicating, maybe it'll stop cancer cells from replicating. So he went away and did some experiments, and Now, cisplatin, even to this day, 30, 40 years later, it's still one of the prime go-to drugs for the treatment of testicular cancer and other solid tumors. Okay, so this was not rational drug design. Okay, and cisplatin itself causes DNA damage, so it itself is a mutagen. So by having treatment with cisplatin, you're exposing yourself to the likelihood of further mutations to your DNA. All right, so short-term gain and long-term possible consequences. Okay, and if that short-term gain is that you live for longer, then good. So the idea is, with rational drug design, you can design drugs for particular cancers that are less harmful to the individual. So we had a look at some of the occurrence of various tumors and the life expectancy with some of these tumors, and that there are improvements with some of these life expectancies and things, but a lot of this is caused due to changes in diet and hygiene and early detection, and a lot of these things, rather than an actual molecular understanding of what's happening. Okay, so the idea is then, what is rational drug design, and what are people trying to achieve from rational drug design? So with a rational approach to drug design, you need to think about a whole bunch of things. So if this is the plasma membrane of a cell here, and here's our DNA here, and here are our little proteins and things that are happening in the cell. So these are the processes we want to try and inactivate to stop cell growth. Okay, so the molecule, the drug, well, firstly you need to get it into cells, and that's the big question mark. So there's certain characteristics of the drugs that you have to adhere to, whether it be the hydrophobicity of the compound, the size of the compound. So we discuss in the lectures some of those issues around the molecule itself that they're designing. Okay. The next thing is, well, what sort of shape of drug is it? What sort of type of drug is it? And how do you make a choice? So for some enzymes in the cell, they've got catalytic clefts here. They've got little cavities in them that bind a small molecule, which is a biological molecule. So things like ATP is a small molecule, which kinases use to phosphorylate things. There's also steroid hormones. There are transcription factors that bind steroid hormones to drive growth. There's other nucleotides that drive growth. There's lots of small molecules that are known about that play roles in cell biology. So if you can design a molecule that looks like ATP, but it's not ATP, it's an analog of ATP, then you can effectively inactivate the kinase. Okay, and if kinases are important in driving growth, and you can mimic the effect of a small molecule and inactivate the kinase by putting a blocker of some description into the catalytic cavity, then you no longer have the ability of that protein to bind to ATP to transfer the gamma phosphate onto the new protein. All right, so an understanding of the activity of enzymes is important to know how to then design a drug for that. Okay, and we all know that we have X-ray crystallography and NMR structures and stuff. Some proteins, they bind to DNA sequence, but there's lots of binding sites and interaction points between the protein and the DNA sequence. So it's harder to design one small molecule to disrupt a binding surface that's much larger. And also, we have protein-protein interactions, and there's all these points where the proteins specifically bind to each other, and it's harder to disrupt those with small drugs as well, because it's just a larger surface area. Okay, so understanding the properties of the protein are important, and how it works. So this brings us to these concepts of is something druggable, and would you want to drug it in the first place? Is it a good target? So if it's if it's an oncoprotein that's driving growth, or if it's a tumor suppressor. If an oncoprotein is driving growth, then it's a good target, because if you can knock out an oncoprotein, you inactivate the thing that's driving growth. If a tumor suppressor is stopping growth, then why would you knock out something that's stopping growth? It makes no sense, because the whole point of the drug treatment is to try and reduce the growth of the cells. So leave the tumor suppressors alone so that they can halt growth. Okay. Question is, can you design a small drug that's going to enhance the activity of a protein? And I would suggest that that is in the too difficult basket right now. There might be one or two examples where you can that might be the case, but generally speaking, I don't think it's plausible at this stage to enhance the activity of proteins through small molecule interactions. So it's harder to make tumor suppressors better, but it's easier to knock out the function of oncoproteins. And the example we talked about earlier was, for instance, if the oncoprotein has a kinase domain, you can replace the ATP with a small molecule, then that kinase can no longer function. So there's all those sorts of things. The thing with RAS has got it's got a small pocket that binds a guanine nucleotide. Okay, the GDP, the GTP. So technically, you could design molecules to try and interfere with its function, but the general consensus is that it's not a good target because it tends to drive growth, and you don't want to lock these growth switches on. Okay, so you need to know, is it a good target? And oncoproteins make good targets. Tumor suppressors don't make good targets. And that idea of a good target is based on the function of the protein and whether it makes sense to put all this time and effort in to target it. Okay. Once you decide that something is a good target, the question then, is it druggable? Is it druggable? So we've got a whole bunch of oncoproteins which probably are druggable. There are other functions in the cell which may not be druggable, even though they're good targets. So it's a good target, but it hasn't got a catalytic cleft or something. So it's harder to design something to attack it. Okay. There's also which stage in the cancer progression are you trying to target these things? Like DNA repair enzymes. If they've been knocked out, then there's going to be lots of mutations. If you can activate DNA repair, it's probably too late. You can't repair a lot of damage that's done. So there's also the rationale of where you are in the cancer cycle about which sort of things you want to target. So we had those sort of discussions, or at least I went through some of those sorts of issues about druggability and good targets. We also talked about this concept of the fact that a lot of anti-cancer drugs do cause problems in normal cells as well, because it's very hard to distinguish normal cell biology from tumor cell biology. Okay, so that's the perennial problem with drug design. We talked about this idea of we've talked about this earlier in the lectures. We talked about the different kinds of proteins which often occur in different cancers. So there may be so for instance, if you're looking at a population that's that's thought to be, for instance, breast cancer. So you've got lots of a population of a cohort of people who have breast cancer. If you think of breast cancer as a single thing, then when you're designing a drug for that, you might get sort of a low response rate. So it's kind of, you're not quite sure whether it's working or not. If you can break this large population up into subpopulations, and by subpopulations I'm thinking subtypes, people who have the similar cancers. So this is type 1, type 2 and type 3. So the biopsies from these patients are more similar. So it's more likely that these people have a kinase that's been inactivated, for argument's sake, a particular kinase. Whereas in this population, it was scattered. So if you treat the same drug here to this population, you might get a high response rate, which means it's a really good drug. But that high response rate was masked by these populations in here. So you got 1, 2, and 3. Because these are not responsive, it masks the fact that this population was responsive. So you sort of go, I'm not quite sure whether that drug is effective or not. Where if you can subtype that into smaller categories and have an understanding of what's affecting that population, then when you do your drug treatment here, the same drug, then you get a much higher response rate, and that's much more that's going to drive the process through clinical trials and all the expense to actually design a drug, because it's very effective, as opposed to something that appeared to be apparently not very effective. Okay, but the same drug is apparently more effective if you can look at a subtype that's more homogeneous. Okay, so how do you look at whether cancer biopsies are similar to each other or distinct in terms of trying to work out these categories that might exist? And modern technologies such as gene expression arrays are used for that kind of thing. I don't want to get too much into the technique, but I guess what's going on here is that you've got 2 cells, and within those cells you've got gene expression occurring, and that's what microarrays are measuring. They're a measure of gene expression. So you're looking not at the genome content, but at what genes are activated. So looking for messenger RNAs. Okay, so if a particular protein is being really highly expressed in a cancer cell, and it's high, but it's lowly expressed in a normal cell, then maybe that expression is bad. Okay, and you want to know that, and you want to start saying, well, here's a subtype of cancers which have got these genes expressed highly. So the microarray, it works by you those messenger RNAs are labeled to make them fluorescent, and you convert them to DNA, so it becomes sort of a DNA with a fluorescent tag on it. Okay, and then you do the same experiment for the normal cells, and you label up all of the messenger RNAs. So all of the messenger RNAs, might be thousands of them, different types of messenger RNAs, they're all labeled from both cell types. So you're getting these cDNAs, and these are labeled with a blue fluorescent tag, and these are a green fluorescent tag, or sorry, a red fluorescent tag and a green fluorescent tag. All right. You then get some sort of substrate here, some sort of glass slide, and it's got spotted here single-stranded DNA sequences, and each sequence represents the coding region of a gene. And then you simply anneal, you mix these two together and then wash them across the slide. Okay, and then so you wash the slide with a mixture of these fluorescently tagged cDNAs, which are direct copies of the messenger RNAs from each cell type. Okay, and if a particular gene, we know that to be the RAS gene, for argument's sake, if these were colored green, and it's the cancer cell, so if this spot was green, then it would say that gene is highly expressed here. Okay, whereas if this spot here, which might be another protein, it was red, so if this was red, it would say, well, that gene was more expressed because there was more RNA in the normal cell. So looking at the color profile of the dots, each dot refers to a gene in the genome, you can look at the expression level, and that's really important information. Gene expression is a good predictor of whether you've got protein levels of that in the cell. Okay, and if something is orange, so it's a mixture of green and red, give you this orange color. If it's orange, then there's no difference between the two experimental samples. All right, so that's what the gene arrays are. They're just these techniques that we use to identify levels of gene expression for each gene in typically the genome using these microarray slides. And there's other technologies as well around these days which haven't yet made it into the textbooks. So there's an example here where they looked at these lymphoma samples, so B-cell lymphomas, and there's a wide range of different types. And from the different types, so the different types are labeled here. There's 1, 2, 3 types of biopsies of patient categories, if you like, and they can look at whether there's certain genes that are overexpressed or underexpressed in one subtype over another, and then they can then choose to treat them with drugs. So you're not treating all of those patients with the drugs because the drugs would be harmful to some, but the cost-benefit analysis, it's better to take the drug if you have the disease that will respond, rather than taking the drug and having no response. So it's just a way of targeting the drug to the people who are more likely to be responsive. So if you have a better understanding of the subtypes here, and you can see here for the different populations here that they identified different proteins and genes that were present or being expressed, and the viability or the response of the different samples to the drug were very different, and they were able to get a very high response from one subtype to the drug with lower responses from the other subtypes. So if you can do that to actual patients in the actual clinic, and you can subtype what they've got, then there's a much better chance you can treat them with a drug that's going to be effective, and spare them having to be treated with a drug that's ineffective. Okay, so we went through some examples of drugs that have been used in the clinic. There are some successful drugs. How do they work generally? There's a bunch of drugs that have three-dimensional structures, the ability to make cancer cells differentiate. I think all-trans retinoic acid was one of those. And there are some other drugs which are able to I noticed there was a bit of confusion about this, allow to make apoptosis happen. What this is saying, this little diagram here, the cells. It's saying that so they can induce. If you look at the similarity between different tyrosine kinases, so there's a couple of examples given in the lectures of these. So quickly have a look at these tyrosine kinases here, and then there are other kinases. There are I assume that's we start to look at activated what I've already talked about, the calcium activated what sort of things do you want to target in the cells, and are they druggable, are they good targets, and types of kinases, not just what receptor is druggable and what is screening target kinases. So it's looking at how similar the catalytic domain of the pocket is of the different kinases and trying to predict, well, if we know that, then it's likely that these are the kinases that will bind to this particular molecule. And again, I guess then you want to start thinking about how you can design the drug to be more effective towards the particular kinase that you want to inactivate, and try and reduce the non-specific effects, because you don't want to perturb too many cellular processes when you're targeting these molecules. And don't forget, these molecules are expressed in normal cells and cancer cells, but if they're expressed more in cancer cells, or if they're more active in cancer cells, then the logic is that the drugs have more effect in cancer cells. So any ways to target these drugs to cancer cells over normal cells is always advantageous. And we also talked about whether cell lines make good models for humans. There tends to be more mutations in cell lines than there is in humans, and also the idea that we all have different sets of mutations makes the issue a bit more difficult. So not only is the question, do model organisms represent the human condition? The question is also, well, do tissue culture, which is human cells growing in culture, do they also provide good models for human cells? And there are some examples where even tissue culture models themselves don't behave as the same as a human body would. And there's lots of reasons for that. And there's also another complication mentioned, is that some cancer cells, they tend to express these normal proteins which are effectively able to pump the drugs out of cells. So these small molecules tend to be pumped back out of the cells. So even though they're effective whilst in the cell, they don't stay in the cell long enough, and then they're pumped out, and therefore it's difficult to get the drug present inside the cell. And then again, it's just looking at some of these different kinases and as targets to some of the successful drugs that have been designed, such as Gleevec and some other ones. And then just lastly, I guess if you go back to the first lecture, remember we're looking at villi, and we're looking at these transient amplified cells which are derived from stem cells. And I included that slide because I knew later on in the lecture series there was this discussion in the textbook about some drugs are effective against the transiently amplified cells, but the cancer stem cells are inaccessible to the drug. So whilst you initially have a good response rate in killing off these transient amplified cells, after a period of months, these stem cells can be reactivated to then produce more of the cells that contain the mutation, because the mutation is present in the cancer stem cells, okay, and it's just copied into the transient amplified cells. So there's a bit of complication in the literature that sometimes you can kill the cancer stem cells, which will stop producing the transient amplified cells. So you don't see much of a change initially, but over time these will die off. So it doesn't look like it was effective, but it actually was. And the converse situation would be, well, you kill off these transient amplified cells, but because the stem cells are inaccessible, then these stem cells will produce more cancer cells later on down the track, so you get the cancer coming back. So there's lots of complications with drug design, and it's worthwhile discussing some of those in relation to cancer subtypes, where the cells are in the body, and the rational drug design. Do you want to drug it, and is it druggable? Okay, so there's lots of issues there. Okay, well, hopefully that's some food for thought. Hopefully quickly working through the lectures as I have has been beneficial to you. Like I said, I don't think listening to this lecture in isolation will be particularly good as a study tool, but once you've been through the actual course content, I think if you go through this lecture, you should be able to listen to what I'm saying and go, oh yeah, I remember that, I know what he's talking about. Oh yes, that makes sense to me. And if it doesn't make sense, then you go, oh, well actually maybe I don't understand the lecture content, maybe I should go back to the lecture content and apply some more revision to that area. Okay, so there's only 5 lectures, it's 10 hours of content. It's not too much to take on board, so good luck with processing that, of working through that content. Good luck. All right, thank you, bye.